everyone, welcome to the next episode of the Bio-Inorganic Chemistry course. We have already seen two electron transport proteins, that is the blue copper proteins and iron sulfur proteins. Next, we are going to see the other category of electron transport proteins, that is the cytochromes. Cytochromes are very important electron transfer proteins which are particularly the redox intermediates. Let's see what's the meaning of the word cytochromes. We know that the word cyto means cell and chrome means color. That is, they are colored electron transfer reagents. These cytochromes are actually heme proteins and they act as one electron carrier by shuttling between iron 2 and iron 3 at their active site and are mainly involved in photosynthesis. We can see that cytochromes are heme proteins and they are electron carriers that act by shuttling electrons between iron 2 and iron 3 and they function particularly in photosynthesis. Cytochromes contain the active center as the heme group. We know that heme is porphyrin ring system chelated to the iron atom. Cytochromes are found in all aerobic forms of life. They are found in the mitochondrial inner membrane, also found in the endoplasmic reticulum of eukaryotes, and most importantly, they are found in the chloroplasts of plants and in photosynthetic microorganisms. Moving on, iron functions well in the cytochromes because it contains iron 2 plus and iron 3 plus redox coupled EMF and has a value of the appropriate range for the life processes. Now we move on to the classification of cytochromes. It was first identified in 1884 by Macmann as respiratory pigments. Almost 50 cytochromes are known and their classification is very complex because they differ in organism to organism and even it is quite different within the same species itself. So, each type of cytochromes in its reduced state has 3 absorption bands in the visible range. So, this is an important characterization of the cytochromes, the UV visible spectroscopy. The longest wavelength band near the 600 nanometer corresponds to the type A cytochrome. The absorption of cytochromes in 560 nanometer is type B cytochrome and type C cytochromes has absorption maximum at 550 nanometer. Here you can see the absorption spectrum of cytochrome C reduced state and oxidized state. So, within each class we have different types of cytochromes, that is cytochrome A, cytochrome B, cytochrome C and all are numbered consecutively and we have further designation as cytochrome C1, cytochrome C2 etc. Sometimes the reduced state R band maximum is suffixed and it is given as cytochrome C559. So, that is the UV R band maximum value and we name it accordingly. Now, we are moving on to the classification of cytochromes. Cytochromes are classified based upon the type of heme present and its mode of binding. Cytochromes are being classified as cytochrome A, cytochrome B, cytochrome C, etc.
Now let's look into the type of substitution and linkage in these cytochromes.
Cytochrome A has formyl group as the substituent in heme. Cytochrome B is covalently bonded to heme. Cytochrome B, protein is not covalently bonded to heme. In cytochrome C, it has covalent links between the heme and the protein. In cytochrome D, we have dihydroporphyrin group. So, mainly cytochrome A, B and C are very important. In cytochrome B, it is not covalently bonded, and in cytochrome C, it is covalently bonded between heme and protein. Now, this figure depicts the bonding and the type of different hemes present in the cytochromes. So, depending upon which type of heme is present in the cytochrome, they are classified as cytochrome A, B and C. In cytochrome A, we have heme A. So, here you can see heme A, it has the substituent different from the other two. Here, this is cytochrome C will have heme C, and you can see the substituent is different here. And this is the cytochrome B, which is present in hemoglobin, myoglobin and cytochrome B have the same heme group. We will learn the structure of hemoglobin, myoglobin in the coming lectures. So, you please understand that depending upon which type of heme, whether it is A, B or C, cytochromes are being designated as cytochromes A, B and C. Here you can see that in heme A, the R1 substituent is hydroxyethyl farnesyl group. The substituent R2 in heme A is CH double bond CH2. The R3 substitution in heme A is the aldehyde group. In heme B, you can see that both R1 and R2 are the same, that is CH double bond CH2, and the R3 substituent in heme B is CH3. In heme C, the substituent is entirely different, and the R3 substituent is methyl group in the heme C. Here you can see the subclasses of cytochromes. We basically say we have cytochrome A, B and C. It is further divided into cytochrome A1, A2, A3 depending upon in very minute differences. And all these are part of either photosynthetic chain, or they are all either independently or in cooperative mechanism are part of different life processes. We have already seen that cytochrome A will contain the prosthetic group heme A, the cytochrome B will contain the prosthetic group heme B, cytochrome C will contain the heme C, and D contain heme D. There is no known cytochrome E, but we have cytochrome F, which is found in cytochrome B6F complex in plants, and it is a C-type cytochrome. We can see that we do not have a cytochrome E, but we have an F cytochrome which is part of the B6F complex. What is B6F complex? We will see in the coming slides. In mitochondria and chloroplasts, these cytochromes are often combined in the electron transport chain or in the metabolic pathways. We can see that cytochromes subclasses are being functioning as a complex. You can see that cytochrome A and cytochrome A3 together form cytochrome C oxidase, which is known as complex 4. We can see that cytochrome B and cytochrome C1 combinedly form coenzyme Q, cytochrome C reductase, which is known as complex 3. Similarly, cytochrome B6 and cytochrome F combine to form the plastoquinol plastocyanin reductase. Distinct family of cytochromes we have cytochrome P450. This is another class of cytochrome. It is named quite differently. This is named as cytochrome P450 because of its particular characteristic, that is, it has a solid peak which is formed by absorbance of light at wavelength 450 nanometer. So, it is given the name cytochrome P450. The solid peak means it is a sharp peak in the blue region of the solar spectrum. Now, we are going to see the structure of the cytochromes. First, we can see that cytochromes B and C have two strong axial ligands and they are low spin. They are 6 coordinated. You can see the picture of cytochrome C. It is 6 coordinated and it is having a low spin. So, unlike the hemes that is present for oxygen transport, that is hemoglobin, it is having no position for further coordination. That means it is hexa coordinated and there is no space for further coordination of any other group. In hemoglobin, we will see in the further lectures that hemoglobin, deoxyhemoglobin is actually pentacoordinated and it has a site for the binding of dioxygen. But in case of cytochrome, you do not have any further vacancies, and since it is 6 coordinated, it is completely coordinated. So, it cannot act by coordinating any particular functional groups or any groups, but they act as electron transport carriers, or they will act as electron transfer mechanism. They act through electron transfer mechanism, or they function through electron transfer mechanism. Since it is completely coordinated, it is 6 coordinated, so there is no further vacancies. So, they function by electron transfer mechanism. Now, we are going to learn about cytochrome C in detail. It is one of the very important cytochromes present in living systems. It is actually isolated from mitochondrial membranes. Now, we will see what is the function of cytochrome C. Cytochrome C actually aids the reduction of oxygen and transmits its oxidizing power
towards burning of food.
It is a widely studied cytochrome. What happens is that the electrons released by the oxidation of carbohydrates are being transported to dioxygen in mitochondrial cells via 20 redox centers involving cytochromes. So, it is a complex process. You should understand that cytochrome C is involved in the oxidation of carbohydrates and they are transporting the dioxygen in mitochondrial cells via 20 redox centers that involves cytochromes. This is the pathway of that. So, the mitochondrial electron transfer primarily involves cytochrome C. Now, let us see the structure of cytochrome C. So, in cytochrome C also we have the main structure, that is the basic heme group, and then it has a polypeptide chain that is attached or wrapped around it. It has a number of amino acids ranging from 103 in certain fishes, 104 in another type of fishes, and in each category of animals, the number of amino acids present that form the cytochrome C is different. It has a nitrogen atom from the histidine segment and a sulfur atom from the methionine segment in this chain, and are coordinated to the 5th and 6th coordination of the iron atom. We know the structure of heme, and you know that for coordination is with the nitrogen atoms, and you have a 5 coordinated space and 6 coordinated space above and below the plane, and these are occupied by the nitrogen atom from histidine and the methionine segment. And there is no further space for coordination. And since there is no further space for coordination, it is of course involved in the electron transfer mechanism. Now, we are going to see how the cytochromes will function.
At the center, there is a metal ion.
It is readily capable of interchanging between the oxidation states. So, that is the active center that will get converted into the next higher oxidation state and will reduce to the next lower oxidation state. For many cytochromes, the metal ion present is iron and it interchanges between the iron 2 plus, which is the reduced state, and get oxidized to iron 3 plus, which is the oxidized state. So, this will get switched Fe2 plus to Fe3 plus and Fe3 plus to Fe2 plus. So, this is how the cytochromes function. Now, we understand that cytochromes are capable of performing oxidation and reduction, and they likewise take part in the electron transfer process. We can modify the cytochromes to meet the needs of the electron transfer scheme. So, in each organism or each life processes, it is organized in such a way that it meets the requirement of that biological processes, and we already learned that it is different from species to species. Many cytochromes acts in combination for specific biological processes. Here, the enzyme cytochrome C oxidase is composed of cytochrome A and cytochrome A3. Similarly, the enzyme cytochrome C reductase is composed of cytochrome B and cytochrome C1. So, the cytochromes in combination will perform specific biological processes. Now, we will learn more about the enzyme cytochrome C oxidase. It is one of the important topics in bioinorganic chemistry. So, I mentioned that it is known as complex 4 and it is a combination of cytochrome A and cytochrome A3. It is the terminal member of electron transport chain. So, we know that the electron transport occurs in a acid chain reaction, and this cytochrome C oxidase is a terminal member of the electron transport chain. So, here you can see the structure of the complex, not exactly, but a depiction of the structure of the cytochrome C oxidase. We will see the function of cytochrome C oxidase. Now, it catalyzes the reduction of dioxygen. What is the function of cytochrome C oxidase? It catalyzes the 4 electron reduction of dioxygen. You can see that oxygen is being reduced by utilizing 4 electrons to water. That is the function of this complex 4 or the cytochrome C oxidase. You can see that it is composed of the metal copper and you have a heme unit, and this is the cytochrome A and this is the cytochrome A3. It is in this cytochrome A3 that the oxygen gets bind and it is getting reduced. We will see the mechanism in the further slides. So, you should understand that this enzyme cytochrome C oxidase has 2 copper sites, that is copper A and copper B. One copper A, that is one copper atom A, and a heme, that is the cytochrome A contains heme, participate in the electron transport process. And the next one, that is the next heme, that is cytochrome A3, and the second copper will combine and function as the reduction or will function for the reduction of oxygen to water. So, this is the mechanism. We have 2 parts, cytochrome A and A3 in combination forms this complex 4. So, one part, that is cytochrome A and copper A, will participate in electron transfer process, and the next one, that is cytochrome A3, and the second copper B, will function for the reduction of oxygen to water. That means it has 2 iron heme centers and 2 copper 2 centers, and it is involved in conversion to or the reduction of oxygen to water and for the electron transfer processes. So, after the process, iron 2 is getting converted into iron 3 heme and 2 copper 2 plus, and what happens to oxygen? Oxygen will take 4 protons and it will take 4 electrons also and get reduced and get converted into water. We have already learned the structure. I hope it is very clear for you that in this section you have cytochrome A and which contains heme A and copper A, and in this section you have cytochrome A3 that is containing heme A itself, and together you have the next copper atom. In cytochrome A3 it is pentacoordinated and you will have space for binding of oxygen. But in cytochrome A, the problem is that it does not have pentacoordinated, or it is completely coordinated or 6 coordinated. So, you do not have any space for the binding of dioxygen. So, we say that this part will function for the oxygen reduction, and this will involve only in the electron transport mechanism. So, this is the structure of the complex cytochrome C oxidase. Next, we will see how cytochrome C oxidase enzyme is involved in cyanide poisoning. The cytochrome C oxidase is actually responsible for unusually severe and rapid toxicity of cyanide ion. We have heard about cyanide poisoning with hemoglobin. So, in that case, what happens is that cyanide ion will bind with hemoglobin at the dioxygen site, and it will bind so strongly that iron 3 hemoglobin gets converted into, or iron 3 it gets converted to iron 3, that is known as methemoglobin. Actually, this is not a common phenomenon. However, the bond of cyanide to the hemoglobin is very strong. Even though it is methemoglobin is formed, it will be present only in few amounts. So, by taking medicines like amyl nitrate or sodium nitrate, we can oxidize some of the
hemoglobin to methemoglobin. The methemoglobin is useless for dioxygen transport, but it binds cyanide even more tightly than hemoglobin or cytochrome C oxidase and removes the cyanide poison from the system.
But when it comes to cytochrome C oxidase, what will happen is that it will bind at the 6th position and it will stabilize the iron 3 so that there is no taking part in the further electron shuttle. So, this is more common to occur and even more severe and dangerous than the hemoglobin poisoning. So, it is very severe that when cyanide ion binds with cytochrome C oxidase, there is the stoppage of the electron shuttle mechanism and it causes severe cyanide poisoning. Now, we move on to the next cytochrome category, that is the cytochrome B6F complex. So, this is another cytochrome based enzyme which is found in plants, cyanobacteria and green algae. They are also found in the thylakoid membrane of the chloroplast and it is part of the photosynthesis. So, what is the function of cytochrome B6 complex? It transfers electrons from plastoquinone to plastocyanin during the light reaction of the photosynthesis. We will see what is the light reaction, what is the mechanism or the chemistry of photosynthesis in the coming lectures. So, you please understand that this enzyme cytochrome B6F complex is part of the light reaction and photosynthesis. So what is the purpose or function? It transfers protons from the stroma of chloroplast to the lumen across the thylakoid membrane. Thus, the electron transport via cytochrome B6F is indirectly responsible for creating a proton gradient that will drive the synthesis of ATP in chloroplast during photosynthesis. So, this is the diagram. Here you can see the B6F complex, which is part of the complex chemistry of photosynthesis. We will learn it in detail in the further lectures. Now, we are going to see the next cytochrome category, that is cytochrome P450. Cytochrome P450 is present in animals, bacteria and plants. It belongs to a family of enzymes having heme as the cofactor. Here also we have heme as the cofactor. So, what does it perform? It catalyzes the introduction of oxygen atom from an oxygen molecule to the substrate. So, this is oxygen molecule O2. It catalyzes the introduction of oxygen atom from oxygen molecule to a substrate, or we can simply say it catalyzes the hydroxylation process. That is, it introduces an oxygen atom from oxygen to the substrate. So, that is the function of cytochrome P450. It is present in mammals and it helps in the oxidation of steroids, fatty acids, xenobiotics and participate in biosynthesis. So, I already mentioned that it is part of the hydroxylation process, and what does it do? The cytochrome P450 enzyme convert xenobiotics to the hydrophilic derivatives which can be readily excreted. So, we can simply say that cytochrome P450 are the terminal oxidase enzymes in the electron transport chain. It is the terminal electron oxidase in the electron transport chain. We already studied that it is called cytochrome P450 because its absorption maximum occur at 450 nanometer. Most cytochromes P450 require a protein partner to deliver one or more electrons to reduce the ion and eventually reduce dioxygen. The structure of cytochrome P450 contains a heme ion
as the active center,
and also it is tethered to the protein via cysteine thiolate ligand. Now, moving on to the summary. In this module, we have studied what are cytochromes. Cytochromes are simply the colored electron transfer agents that are part of the electron transport chain, and these are the redox intermediates. In most of the cytochromes, we have heme group as the active center. They are found in the mitochondrial membranes and chloroplasts of plants. We have learnt about cytochrome C oxidase, the function of cytochrome C oxidase. We have studied about cyanide poisoning. We have also learnt about the cytochrome B6F complex and also cytochrome P450. Thank you.